Hello, I am Ashlesh Pattanaik and I will be talking about a part of my master's project on acoustic partitioning between two sympatric tree cricket species. So, sympatric speciation is the reproductive isolation of two closely related species without any geographical barrier. So, they adopt various partitioning strategies to prevent heterospecific mating. In case of acoustically active organisms, these strategies can help in avoiding song interference. So the different species can occupy different vertical strata in a forest, or they can choose to call in different seasons or during different times of the day. And if they do call at the same time, then they can have unique features to their calls that can only be identified by their own species. This is called acoustic partitioning. So we wanted to identify the call feature or features responsible for species identification in each of the two sympatric tree cricket species. Now closely listen to this audio where you can hear two different call types simultaneously. So the first call was from Oecanthus indicus as shown here, and the second call is from rufescens as shown here. So as we can see, rufescens has a continuous trill while indicus calls in chirps. And the other difference is that the carrier frequency of rufescens is almost a kilohertz higher than that of indicus. So we need to find out which feature is the best for identification. So the methods include field sampling, recording the male calls, collecting and rearing the females. Then the calls were analyzed and different call types were synthesized. And this was followed by playback experiments where the female has to move towards or away from the speakers playing the calls. Now the calls were designed to look at one feature at a time. So let's say broadly the call has three features: carrier frequency, call duration, and intercall interval. In nature, these features can have a minimum, mean, and maximum values, and this is shown here by the light, medium, and dark colors as well as the numbers in the brackets. So these are the conspecific calls, the blue ones for rufescens and the orange ones for indicus. Now we prepared some hybrid calls. So let's say we are playing calls for the rufescens female and we want to test the importance of carrier frequency. So we replaced its natural carrier frequency by that of indicus, whereas the rest of the call remains the same. To test whether it responds to different values of the frequency of indicus, we used different treatments as F1 or minimum, F or mean, and F3 or the maximum value. Similarly, we can test for other features such as call duration and intercall interval. Now, this is a typical phonotaxis event where the female moves towards the playing speaker. So for each experiment, we gave 5 treatments to each of the females. So the conspecific call, the 3 hybrid calls, and a no-call scenario as a control. So here we can see that 8 out of 10 females have responded positively to the natural conspecific call, whereas there were fewer responses to the hybrid treatments. So this was found to be statistically significant using the McNemar's chi-square test. And for call duration and intercall interval, the difference between the conspecific and the hybrid treatments were not very significant, showing that the rufescens females can respond to indicus calls with respect to these two features. On the other hand, indicus had 7 out of 10 females responding to the conspecific call, but due to the 3 positive responses to this particular hybrid, this was not very significant. Thus, it is inconclusive whether indicus can also respond primarily to its conspecific frequency or not. I would say that the call duration and intercall interval for indicus, the experiments are yet to be done, hence they are not shown here. So from these preliminary experiments, we can say that there's a possibility of frequency partitioning as at least one of the species responds strongly to its conspecific frequency, while the macro-temporal features such as call duration and intercall interval might not be very important for species recognition. In future, we will be increasing the sample size to have a more robust conclusion. Now, literature suggests that micro-temporal features such as syllable period and duration play an important role in species identification. So we definitely need to test those in the future as well. And apart from that, we also need to test the interaction of 2 or more features and if they have some impact on recognition. I would like to thank my supervisors Roy and Rittik for helping me out with this project and my wonderful lab mates for their support. I also thank ABL for giving me a platform to present my work. Thank you all.